speaker is Professor Kandice. She's a biophysicist, and she originally comes from Trinidad and Tobago, which is a small country north of Venezuela. And Kandice did her PhD in physics at the University of Illinois in Urbana-Champaign. She went for a postdoc at the University of California, Irvine, and then at another postdoc at the University of California, Berkeley, and Lawrence Berkeley National Lab. In 2012, she became a Stedman tenure track investigator at the National Cancer Institute in the US. And in 2020, she received tenure at the National Institute of Health in the US. And she is now the head of the tissue morphodynamics section at the National Institute of Health. She received many awards and honors, and I want to mention a few. In 2013, she got the National Cancer Institute Directors Intramural Innovation Award. In 2015, she got the NCI Leading Diversity Award, and in 2016 and 2018, she got the Federal Technology Transfer Award. She currently serves on the Committee for Inclusion and Diversity of the US Biophysical Society. Her research focuses on the investigation of metastatic traits that allow tumor cells to colonize secondary organs. And this is also the thing that she will present us today. The title of her talk is Microenvironment Regulation of Metastasis. So Kandice, the stage is yours. Go on.
So thank you very much for the opportunity to share our research today. And we're going to try to bring a different perspective on how you could use tools and approaches that are employed by other fields of physics to be able to address a very important question of, unfortunately, of clinical need. So as mentioned, my title is looking at cues from the microenvironment and how they regulate a very specific stage of cancer known as metastasis. So we still need to define these terms so that we are on the same page. So before I play this movie, metastasis simply describes the process by which cancer cells here, shown in purple, are able to leave a primary organ, enter into different conduits, different tissues, such as blood vessels, and then be able to survive in these conduits, exit, and unfortunately form new lesions. It still is a great cause of clinical lethality, so it's still of great clinical importance to study. So when I play this movie, one thing that we've learned over the last 50 years or so, that genetic expression of these tumor cells are important in driving these processes. How these cells receive cues from the environment via chemokines are also important. But if we focus on this movie here as it loops, one thing you could appreciate is that the cells undergo different types of morphological changes. So they change shape. They encounter other types of cells, and they have to go through different physical barriers to be able to establish these lesions. So when we first started the lab, the question we had is, could we bring some insight in terms of how these cells sense these different environmental cues? How does that then govern the site of these secondary lesions once these tumor cells have left? And once these tumor cells have gone to these distant sites, how are they then able to grow in an environment that's completely distinct from the one that they originated in? So I described to you what metastasis is, but let's talk about what I mean by microenvironment. So in this schematic here, I'm using cutaneous melanoma as an example. And within this model system, melanoma cells can interact with other melanoma cells, interact with other matrix components or cell types. But one thing I want to introduce to you is that the tissue properties can be defined in just as they were materials, living materials, in that they could have viscoelastic properties, in that the way that these extracellular matrix proteins are arranged can convey this sort of physical cue. But yet these cancer cells are malleable, such that when they go to a distinct organ, and in this example, I'm using the brain as the example, we see that not only are the cell types different, but the different cues that these cells receive in terms of physical properties are distinct at different length scales. So we'll talk a bit more about that. So how do we then model these complex systems in that? How do cells interact with each other? How do cells interact with other cell types in the environment? One thing I want to put out to you is that cells don't just sit there statically. They can generate forces and impart them on a local environment. I'm going to show you 2 examples. On your left here, you're looking at breast epithelial cells. So these are the cells that form ducts within breast of mammals. And we're placing these cells in an environment, a hydrogel, that mimics both the chemistry as well as the physical properties that it would see in a native human environment. And here we see that these cells, in order to form these structures, undergo this sort of coherent rotation and therefore communicate with each other in these cooperative behaviors. In a similar vein, if we put a different type of cell here in a matrix, collagen type 1, which is found one of the most abundant proteins found, extracellular matrix proteins found in the body, you could see that this cell is using protrusions that are then regulated by a key protein known as actin to generate contractile forces. And you can see that there's some deformation in the underlying matrix. So right away, we see that even when we look at these processes, we also have to investigate at different length scales, temporal scales, and I'm going to introduce you some force scales. So when we first started this, some of the things we wanted to achieve is being able to understand and not shy away from the complexity of what cells may see in a native in vivo environment. We want to be able to not just visualize it, but try to bring some mechanistic insight, bridge the physics with the biology. And these are 2 lateral things that we'd like to focus on, but today, in the interest of time, I'm not going to talk to you about. So there's one more thing I need to tell you about how do we study this. I've introduced some model systems before where we could use these 3-dimensional hydrogels. Again, these are usually composed of matrix components that mimic chemistries and physical properties that we've measured. Here you're looking at a cell interacting with its matrix in this black box. And the cell is able to grow and move. And we could use the system to understand cell interactions with its environment. In order to build in the complexity, because one thing you could notice in terms of the hydrogels versus that of what may be found in mammalian systems is the ability to look at blood vessels. I mentioned that this is a conduit by which tumor cells can migrate within to form these and escape to form these new lesions. So I'm going to talk to you a bit more about this today. And finally, we also look at tumors in different animal models that allow us to not only look at these single cell dynamics, but tumor dynamics and do so in multiple organs. So in the interest of time, I'm just going to talk to you today about this study and how do we then incorporate this model system to understand how biophysical properties may regulate metastasis. Okay, so let me introduce what you're looking at. In this big micrograph, this is the head of a larval zebrafish. It's 3 days old, so 3 days post-fertilization, 1 day post-injection. And this is his eye, this forebrain, midbrain, hindbrain. It's a transgenic line that allows you to look at blood vessels, as well as the neutrophils, which is a type of immune cell for defense. And in blue, we have human cancer cells that we've introduced directly into circulation. One of the beauties about using the zebrafish, it allows us to visualize these sort of cancer dynamics that are not easily observed in mice models. And here you could see at 5 days after these cells were introduced that some of these tumor cells have divided, some of them have not, but more importantly, we see this diversity of behavior with respect to the microenvironment, in this case, blood vessels. So when I play this movie again, if we look at the big micrograph, one can appreciate that there's a diversity in types of behaviors. Some cells have a round morphology, some cells are more elongated. And if we go into the inserts, we could see that the cell has successfully extravasated and is now surfing along the blood vessels. Whereas in the top insert, we could focus on the fact that not only are these cells left, they're able to extravasate, and we catch that act, which is very difficult to get in other systems. And just to give you an idea of timing, it takes about 30 minutes for this to happen. So it's a really fast process. So now that I've introduced a model system that allows us to be able to address what these cells may do and visualize them with single cell resolution, the question then is one more thing I need to tell you about metastasis. Metastasis is non-random. So when I first started, I assumed that cancer cells had some probability of escaping. It's a purely stochastic process, and some probability of surviving, some probability of getting out, and that there should not be any bias of one organ versus the other. But indeed, that's not the case. Instead, cells have a preferential secondary site of colonization based on where these cells originated from. So first likened as seeds in a hypothesis put forward in the 19th century, it's now been put forward that if some cells have this intrinsic property, only if they fell on congenial or soil that's compatible with that, then you have a successful outgrowth. I'm showing you here for 4 examples. So prostate cancer, some subtypes show a preferential colonization of the bone, whereas lung and breast cancers show a more promiscuous behavior, colonizing distant organs, albeit with different latencies, as given by these arrows. So there's one level of complexity that we need to build in to make sure that our model system at least closely resembles what may happen in human disease. So returning to our original question, what governs the selection of secondary site? Why do we then see this non-random behavior? So the strategy is as follows. First, we have to show that using our zebrafish model, that we could recreate this aspect of non-random organ selectivity. I talked to you a bit about how cells talk to the microenvironment, but I didn't talk to you about what biophysical cues are relevant and how do we then measure them in vivo in a living animal. And finally, by exploiting this information, could we then use some genetic information to drive differences in how these cells go to these different organs? Okay, so first, we have cell lines that when you put them into mice, they either go to one organ, such as the brain or the bone marrow. These are breast cancer cells. And we simply ask if these same cells will show a similar selection process where they will go to the zebrafish brain versus that of the zebrafish bone marrow at these ages. So we inject them into the fish at 2 days old, and we simply quantify this process. So what did we see? On your left, we're looking at now the heads of the fish. This is now the eye, fore, mid, hindbrain. This is 5 days post-injection. And you could see that for cells at home to the brain of the mice, they also home to the brain of the fish. What happens when we inject this clone? What we observe is that, again, this is the eye of the fish, that when we inject this clone that goes to the bone marrow, we do not observe colonization or extravasation. But that does not prove that there's non-random selection. But because we have single cell resolution, we could look at the bone marrow niche of each of these fish. And what we observed is while brain targeting cell will go to the bone marrow niche or cortical plexus in this model, we see that the cells don't survive and they're largely dead, whereas the cells that home to the bone marrow are now also found in these bone marrow niches where they have extravasated and proliferated. It's very important for us to validate our model system with other cell lines, and I could assure you that this was confirmed for other isogenic pairs as well as cell lines that show this distinct colonization preference. So now that we've discovered that we could use the system because it has sufficient homology, let's define the biophysical properties. So this is an extremely busy slide, so let me walk you through it. It has been shown that the relative sizes and architectures of the conduits, in this case a blood vessel, can regulate how cells migrate, how they generate forces, and more importantly, how they govern cell fate decisions. On your right, we need to define, I mentioned that tissue has material properties that can be defined by viscoelastic formulae. And here we are simply showing that we can do so safely in a living zebrafish. So we developed an optical tweezer system such that we could apply near-infrared light. And here you're looking at us just simply using our trapping force to be able to move a blood cell within a living zebrafish at 500 microns. And this allows us then to define that we could safely interrogate a living animal. And more importantly, we have the signal to noise ratio to pull out what the viscoelastic properties are. And I'll talk more about how we do that in successive slides. Finally, we're putting cells within a living animal where blood flow is undergoing. So we need to be able to measure the contributions due to blood flow and delineate those from that of migration. So we need to go fast enough to be able to differentiate these different flow rates. And here we use microfluid devices as a control. And we have put in human cells in zebrafish. And while there's excellent homology, we've demonstrated that there's this non-random selection conserved. The idea that there is some difference, there are differences, and we then mimic what these cells would see in vivo using these in vitro devices, but then we put human proteins in there as a comparison. So let's go through quickly. So going back to what metastasis is, I told you the dissemination of cells from a primary site, it enters into the conduits and they're able to migrate, they leave, and then they survive. We're not going to be able to look at the first 2 points. Okay, so the question is, why do we then observe these differences? Is it that these cells migrated differently within these conduits? So first, let's quantify what types of architectures these cells are seeing. So to remind you, this is a larval zebrafish. This is its head, the brain, the cortovascular plexus, which is our bone marrow niche. And when we quantify the relative vessel widths at this age at which we see extravasation events, we see that this is comparable to what you would observe in a capillary lung of a human. So what I want you to think at this stage of using this model system, that it can be likened to a human capillary bed. And that's going to become important in a sec. Now, the fact that we are putting cells in fish that are migrating at the same time. So we need to do some image registration to make sure that we could really truly answer this question of, do cells show different migratory patterns in vivo? So once we've done this sort of correction, we could then use our microfluid devices and then ask, do brain-targeted cells migrate differently or preferentially go to the brain? And that's why we saw that non-random selection, or is it other factors? So using these methods now, we could then tell you that no, there were no differences in terms of migration patterns. I'm showing you here average speeds. We looked at persistence and directionality, independent of in vivo, in vitro, independent of what type of matrix. You do see differences if you change the geometry to two-dimensional substrates. So in our hands, it's not that there's a difference in migration strategies, accumulation, or interactions. It simply is that these cells could equally go to each of these organs within circulation. So then we revisit our canonical explanation of metastasis, and we ask, are there differences? Maybe once they get out, the reason why we see this non-random is that there are differences in survival. So we injected them directly into the organ of interest. And to cut to the chase, the answer is no, in that we see equal survival. So it points to the other point here in terms of extravasation may be the rate limiting step. And this is what we think based on some vignettes I'm going to share with you. So remember I told you that the zebrafish at this age, at least architecturally, can be likened to massive capillary beds of what is found in human organs. And this is important because based on some literature research and others, in that the cell's ability to get stuck in these organs are usually in the capillary beds. And here on the right, I'm showing you now a micrograph where the human cancer cells are in blue. And this is now the zebrafish endothelial cells that are tightly regulate the cells' interactions. So you could see that they're hugging. So we decided to focus on this interface to see what cues they receive in terms of driving this non-random selection. So the first thing we ask is, because mechanical properties have been shown to be important in directing cell fate behaviors, are these mechanical properties the same in the brain endothelium versus that of this bone marrow niche? This I'm just giving you a sort of snapshot of mechanobiology, is the idea that cells and tissues can be treated with material properties. Cells themselves can generate forces on the order of piconewtons to nanonewtons, and then they can remodel matrices on the order of hundreds of microns that then in turn change tissue mechanical properties on the order of millimeters. But yet this process has been shown not just a physical way to describe the material properties of cells and tissue, but they can directly influence things such as gene expression, cell migration, which then regulate how cells behave and cell fate decisions. Okay. So here, I'm going to, I should probably take off the laser here. So here I'm going to show you how we attempt to do this system here. So we have these beads that serve as our mechanical sensors, and then we apply forces. I'm sorry, that should play. Give me a sec. All right. I'm gonna go back. Cannot play media, okay. This should play, I apologize. So what we do is that we apply we use our optical tweezer setup here, and we do a frequency sweep about 3 hertz to 15 kilohertz here. And just for the aficionados, it takes about 90 seconds for us to be able to do these measurements. We're able to then use this as our mechanical sensor to map the mechanical properties of the underlying tissue. And we do so about 20 frequencies, 20 nanometers per frequency. And with some being able to infer from the underlying deformation with appropriate calibration of trap force, then we'd be able to understand what the local mechanical properties are. So what did we find? I'm just showing you the complex modulus of the different tissues, and focusing on this graph here. One thing I just want you to show as a function of the y-axis, increasing values show that the material is stiffer. On the x-axis is this frequency sweep to really do this sort of frequency-dependent motion into frequency-dependent modulus. And what I want you to observe is that for the ages, 48 hours, so 2 days old, for both the endothelium at the brain, the endothelial interface at the brain, as well as the cortovascular plexus, they're largely the same in terms of this complex modulus. Now, I didn't show you now in terms of the loss tan delta to be able to appreciate how solid or liquid-like the material is. And more importantly, they are largely the same, unfortunately, for us. So it's not that mechanical properties are not different, it's just that we can't say that they were unique in terms of driving this in an organ-specific manner. Just to confirm that we are sensitive to changes, we look at the endothelium interface at later ages, at 96 hours post-fertilization, and we see that the bone marrow niche or cortovascular plexus is indeed stiffer than that of the brain at this age, indicating that at least our technique is sensitive to differences in age-related mechanical changes. So I talked to you about mechanical properties. We discovered, at least in this age, that there are no organ-specific ones. But one other factor that can regulate this non-random selection is architecture. And here I'm showing you a micrograph of what a bone looks like. And if you could appreciate from the fluorescence images that these blood vessels can undergo more like a fractured or tortuous vessel organization. And when we revisit our zebrafish model, we see, again, this is the head, this is the cortovascular plexus. We also observe that there are differences in heterogeneities in the vascular organization. So we decided then to investigate that from the perspective of what does a cell see? Again, going that this can influence how cells migrate or receive cues from the environment. So we use micrographs obtained from high-resolution images of our blood vessels in these organs, and we simply calculate the order parameter based on these eigenvalues of using this tensor formulation, such that if it is that the cell on a diameter of a cell sees an environment that's more linear, that would trend to values closer to 1 versus that of a more chaotic or tortuous environment that will go close to the 0. So while the mechanical properties may not differ, what we see, at least on that length scale, is that the cortovascular plexus is indeed more chaotic than that of the brain, and these intersegmental vessels serve as sort of like a control. So the question we ask then is, are there differences in blood flow? And suffice to say, at least at the ages at which we see extravasation, there were no differences in flow in terms of turbulence and so forth. So while flow may be an important factor in driving these metastases, it's not driving it in an organ-specific manner in the zebrafish. So we then ask, is it then the architecture itself? Could that serve as a passive regulator of this intravasation process? So what we did is we first calculated how many cells are present, 12 hours. In the first 12 hours post-injection. And then we calculate how many of them remain stuck in one organ versus the other. So on the y-axis, this is the number of cells that become stuck after 12 hours and how many cells are able to pass through these different organs of interest. On the x-axis, we're using these two different pairs of cell lines that I mentioned before, and then we're using 10 micron diameter beads as our control. And with the caveat that these beads are carboxylated, they should bind equally in each organ. There should not be a bias, whereas these cells that home to one organ versus the other may have different receptors to be able to receive these cues. So what did we find? We found that independently of the type of cell or the 10 micron beads as a control, that they prefer to get stuck in this chaotic cortovascular plexus. But I showed you earlier that the brain targeting cells prefer to go to the brain and extravasated there, and the bone marrow cells then prefer to go to the cortovascular plexus and prefer to extravasate here. So why are we then observing these differences that are just simply due to the architecture? And it goes back to this sort of dynamics of mechanobiology that I mentioned earlier on, is that while we can define these cues,
5 minutes.
Almost done. While we could define these cues, we need to make sure that cells have the machinery to be able to receive them. So if it is that these physical properties are present, but the cells don't have a receptor or a binding partner that allows you to receive these cues, the cells are largely blind to them. So for that, we then examined what types of receptors, what type of machinery do these cells have, and how are they expressed with respect to each other. So two of them, beta-1 and unconventional myosin 1B, was upregulated in the bone marrow and conversely upregulated in the brain targeting ones. And then we allowed ourselves to investigate this one. In the interest of time, I'm just going to show you myosin 1B. So it's found in the protrusion. So again, going back to this idea of the cell's ability to sense these cues of the brain targeting, but not found in the bone marrow targeting cells. And when we silence this using genetic means, we see that the cells lose the ability to colonize the brain, moving from this more linear environment to more of the chaotic one simply by modulating this gene expression. Because we want to make sure that this is not a zebrafish specific phenomenon, we also introduced genetically perturbed cells into mice. And we observed that when we silence this gene, we also lose brain metastasis. So I'm hoping to show you today that by using different organ model systems, we could recreate aspects of human disease. And by using these model systems, it allows us to use a suite of tools that can investigate how cells migrate, the types of biophysical cues that may regulate this process. And by being able to visualize these processes, we could see that just the passive blocking of the cells due to the architecture is an important factor. And then by understanding how cells receive these cues using genetic factors, we could then shift the sites of organ colonization in vivo. So there's one last thing I'm going to leave you with as I close out. This micrograph that I'm showing you, these cells have the machinery that could go to the brain. As you could see, many of them are there and they become stuck, but only a subset of them extravasate. So why is that? How is it that even though they may have the machinery, hand waving, there may be some heterogeneity in the expression, but we want to understand what drove this heterogeneity. So for that, we decided to then map the mechanical properties of these cells as they move through in vivo, as well as how they move through in terms of these in vitro systems that allow us to then quickly compare what changes observed as cells deform to go through these differences. So on your y-axis, again, this is the magnitude of the complex modulus. As it increases a stiffer, we do our frequency sweep. And suffice to say, we see that the cells that are able to get through are the ones that are softened significantly compared to the one that remains stuck. More importantly, what we found is that this may be regulated by a protein known as YAP that could then demonstrate why we see these differences in extravasation processes on top of the complexity of having the right machinery. So I will stop there But first, let me thank the people who did the work. The work was done primarily by Colin and Alexis when they were in the lab. Colin was the senior postdoc who led the project. Alexis is the one who started with the zebrafish work and helped greatly before she went off. I should change it. She's graduated. And the last part, what I quickly went over, was done by Norbert Woong-Young-So, who's taken over this project and really pushed it into different directions that we are excited about. So thanks again, and happy to address any questions that you may have.
Thank you very much, Kandice, for this impressive talk. So it's really fascinating.
Thank you. I'm sorry if I went over time.
No, it's perfect. You're perfectly in time.
Okay.
So just right now, I don't see any questions in the
okay,
Q&A.
I'll drink some tea then.
Yeah, so take your time. So maybe so the people from the YouTube channel, you can ask questions, or of course the panelists also can ask questions if you have some. So Rosaire has a question.
Yes, thank you very much for this really very, very impressive talk. Being not in this field at all, I have a question about the fact that you are using this zebrafish as really the system where you are testing all these migrations. Now, the question is, why is this, you work with zebrafish, and what about when you test that in other kind of, in other set of systems, how much does this dynamics changes or you expect to change? So what is so special about using this population?
So, so when we started, we came from the model of using these hydrogel models, and they work really great in terms of recreating aspects, right? But the idea of being able to build in this complexity of blood vessels, immune cells, different oxygen gradients, and so forth can be achieved to some extent using organ-on-a-chip models. Mm-hmm. But we still don't understand like what types of cells, like how many cells would you put in? Would it be a 3-to-1? What's the stoichiometry? How do we then regulate the chemistry? How could we control this? So then, of course, the other extreme has been to try to do these types of systems in mice. But I didn't really get into that with the mouse, why one could do intravital imaging in terms of putting these different imaging windows. And now it's become much more accessible to look at difficult organs like lung and the liver. The reality is that you still are limited by the penetration depth. You can't get the resolution that you need in these thicker organs using current imaging setups. And then while it's feasible to try to have imaging windows on multiple organs simultaneously, I would like to see how somebody gets an animal protocol quickly approved for that, right? And because it's really strenuous for the mouse. Whereas the reason why we use zebrafish is not so much because they're easy to image, which is one of the factors, is that in terms of what defines zebrafish brains are comparable to mammalian systems. They have the blood-brain barrier, they have the different cell types and so forth, right? But more importantly, we can do these multiplex measurements using genetic tools that mimic what you would see in mammalian systems. So we're very careful what is a good overlap we say and what's not we say. But more importantly, it allows us to look systemically at what is going on in these different lesions, right? That you just can't do with a mouse. But your point is well taken in that we don't stand alone on saying that zebrafish is the perfect model. It's a model system that allows you to address some questions.
Mm-hmm. Thank you very much.
Thank you very much. So since there are no more questions from the audience or the panelists, let me ask, so we have a very special format because we have many students maybe in the YouTube and so on, and maybe, yeah, some of them were asking themselves, how did you choose your field of studies? Because you went to biophysics, so was it your first thing that you wanted to do?
So, no, I started off life as a condensed matter physicist. Okay. So how did I end up in biophysics? I think that, one of the things I hope to impress on people is that, one, you could start off one way and end up somewhere else, right? Two, and how did I end up there is that, we focus a lot on tool development in my PhD, and we collaborated a lot. Like I did my PhD spectroscopy in a cat's brain, but we had to collaborate with a neuroscientist who had the model system. And he's the one who, he trained me, but in fact, it was his system. And if he didn't train me, I would just be reduced to come in doing measurements, analyzing, and having no control over the experimental modulations, right? And I decided that I didn't want to be an imaging squirrel monkey. There's nothing wrong with it, but I just didn't want to not have that control, in that I can't do my own sample prep, or I don't understand how it's done correctly. So that's why I decided to go into this direction that doesn't seem linear, because now I can tell you how to do certain biological processes that I would have no clue before. And it influences the way that we design our experiments, so that why are we going to all these efforts to build these systems that could interrogate multiplexed analyses? And then at the end of the day, the biologists will be like, we don't care what you did, because you did it in a system that doesn't matter. Okay, so that's how I ended up there. Sorry for the soliloquy, because I thought this was supposed to remain scientifically.
Yeah. So thank you. We have one question from the audience. This is from Zainab Al-Zayed. Can we study cellular mechanism using ultrasound to better understand the metastasis mechanisms?
Yes, you can. So thank you for that question. And we didn't talk at all today about our efforts to understand the mechanical properties of the cells themselves, right? So ultrasound has been used in terms of defining these principles in vivo. There are other elastography techniques that are being employed right now, such as magnetic resonance elastography. Actually, shout out to Ingal Sack at the Charité. He's doing like really cool work there. Right, so there's this idea that if we could then use and define the biomechanical properties of cancer cells, maybe we could understand metastatic potential and sensitivity to drugs. Now, the question is now, how do we then move some of the in vitro work that we and others have done to be able to then be more predictive? So that is what I would say would be the biggest challenge that we have. We know we could quantify differences in terms of this sort of mechanical phenotype between what's a normal cell and what's a cancer cell, but we cannot yet use that information, at least in my opinion, so let me speak for myself, to predict if we have this, can we then know what types of lesions, if they will, and drug sensitivity.
Okay, then you just answered the question of Barbara Jacek. Do you have some thoughts about how your biomedical work could translate to therapies or preventive measures? So right now, I would say there's at least some preclinical work in the mouse, like for pancreatic cancers, that show that the collagen, a dense collagen layer that forms around these tumors, if it is then you target this, presumably the mechanical properties are well, because it's a stiffer environment, that makes these tumors more sensitive to drug therapies. So it's still going there, but I think we still have some ways to go in terms of predictability, but that's what we're trying to do in the field right now. Okay, then thank you very much for your talk and for answering all those questions.
No problem.
I will pass the chair now to Elena, and